Okay, so let's start. So a very good day to your vector control colleagues from all across the world, and a very warm welcome to this APMEN Tech Talk webinar. Thank you for setting aside time to join our community of vector control specialists that are listening in. We've had apparently more than 500, 518 to be exact, 518 people that registered for this webinar. And it's always particularly gratifying to see such strong representation from the Asia Pacific region, which is our primary target audience. APMEN, as you probably know already, is the Asia Pacific Malaria Elimination Network, APMEN, that supports our 21 member states across Asia Pacific to achieve their malaria elimination objectives. And APMEN works very closely with APALMA, which is the Asia Pacific Leaders Malaria Alliance, and that is also based in Singapore. Our webinar today is going to continue our previous discussions last year on the use of repellent materials against mosquitoes, and also attractant substances for which there is also a major need. And here's some of the background. Ever since the discovery in 1898 that Anopheles mosquitoes are the vectors of malaria, vector control has been our main strategy to combat the disease. I say that again, vector control has been our main strategy to combat malaria. At first, we focused on larval control and house screening, and then came the DDT era, and finally the era of insecticide-treated nets. Contributions of LLINs and also IRS to reduce malaria transmission in the last 2 decades has been enormous. But as we all know, that impact has now stalled, unfortunately. Much of that has to do with insecticide resistance, but very importantly also increased outdoor biting and early evening biting. That's when people are not protected by bed nets. Outdoor and early evening biting is a very serious challenge for the simple reason that we do not have good methods for outdoor vector control. We are not effectively addressing outdoor biting. Many vector control specialists are therefore quietly and increasingly looking at repellents to reduce the biting risk. And I could explain a lot more, but let's wait for the Q&A session to open the discussion and debate. So just briefly, some comments on the webinar process. We will have 3 PowerPoint presentations back to back, each about 10 minutes in length. This will be followed by a question and answer session. Our audience members can submit written questions during the presentations. Please use the question box, not the chat box, please, because we monitor the question box for questions. Please precede your question with the name of the panel member you would like to respond to your question. That is, if you have a preference, of course. So for example, Dr. Danny, and then a follow-up with your question. After the Q&A session, there is a very short poll just to assess your opinion of the webinar. It's very short, probably 1 minute of your time, but it helps us. And I'd be grateful if you can just quickly tick the boxes. Please note that this webinar is being recorded and it will be made available on the APMEN and ORIN websites within the next 24 hours. Details of these sites will be available on your screens, the URLs. So we've got 3 speakers, as I mentioned, and our first speaker today will be Dr. Dani Lucas Barbosa. Dani did a major in chemistry for her BSc degree at the Federal University of Rio de Janeiro, Brazil, and went on for a master's degree in plant sciences at Wageningen University in the Netherlands. She also did a PhD in entomology at Wageningen, cum laude. Dani now works at the University of Zurich, Switzerland, with a group of Niels Verhulst. Her main interest is in functional and chemical ecology. Dani was the senior author of the recent publication in Trends in Parasitology, which was titled Skin Bacterial Volatiles, Propelling the Future of Vector Control. I found that a very fascinating interesting and evocative title. That's why I invited you, Danny. We look forward to your presentation, Danny, and thank you sincerely for agreeing to share your insights with us. So Danny, if you could activate your video and unmute yourself, and please proceed with your presentation. That would really be great. Thank you.
Hello, everybody. Very happy to be here early in the morning for me. Let's see, now you have my presentation screen mode, I believe. So I'm going to tell you what Leo Brack just anticipated for you. I'm going to tell you a little bit about these ideas that we discussed in this paper that was published last year in spring, but also about the project and some of the results we already achieved along the way working on these ideas. But to start, let me first tell you something about how mosquitoes actually find us and other animals. Here I'm going to focus on humans today, but let's just remind ourselves on how mosquitoes locate us. So CO2 is a very important cue that we like to say that as mosquitoes perceive CO2 and CO2 actually activate mosquito seeking behavior much more than a direction cue. It's a more activation of the searching behavior. And then our own body odors are always very important for mosquito to locate us and also to discriminate between us and other hosts. So differentiate humans from cows or other animals, for example. Body heat is also a cue that mosquitoes can perceive, and eventually even our own body flavor, the skin flavor, also will determine whether mosquitoes decide to bite or not to bite us. But much of the odors, and odors are the main cue we think we can actually interfere when we're talking about repelling mosquitoes or keeping them away from us. And much of the odors that are produced by the skin, our own skin, is produced actually by bacteria. So it's our own microbiome that make us smell different from each other and also smell different from each other in terms of mosquito attraction. So you probably all noticed that if you do sports, for example, you don't immediately start smelling bad, but after a while you start smelling bad. And that's because the bacteria is actually degrading the compounds that are in our sweat into compounds that then smell bad. So I want to mention that this project is actually funded by DARPA, the project I'm working on at the moment. And DARPA is the defense agency of the United States. Why DARPA wants to invest in such a project? Well, DARPA wants to basically protect their own soldiers. When they send their soldiers all over the world, they want to protect their soldiers not only from malaria, but also from dengue, chikungunya, and Zika. And that's why they invest in this project for us to produce a probiotic that's based on skin microbiome. So I never thought I would be working for DARPA, but DARPA have been investing in many interesting projects. So for example, DARPA back in 2013 have been invested in messenger RNA technology for vaccines. And today, many people in the world have been vaccinated with a vaccine from Moderna, including myself, against COVID-19. And this was all financed by DARPA back in 2013. And so DARPA now also finances this project that I've been working on. And what is the goal of the project? So the goal of the project is to produce a live biotherapeutic product that can hide human signals and hamper mosquito attraction. So it's to produce a probiotic, a topical probiotic that you would apply on your skin and repel mosquitoes. But why is that? Why do we need a microbial-based repellent? The reason we need a microbial-based repellent, or we think we need a microbial-based repellent, is because the repellents we use today are basically not effective, or they are not effective for more than a few hours. So these compounds that we apply on our skin today, you use as repellents commercially, they evaporate after a few hours. And that's normal because they are volatiles. And they are made for us to be perceived by other insects in this case, but we can smell them exactly because they are volatiles. But this also means that they evaporate quickly, and then there is needed for reapplication over and over again. So what is the difference then when you have a microbial-based repellent? The idea is that the bacteria then would keep producing these odors continuously when applied on your skin. And that actually the bacteria would even reproduce on your skin and therefore continue. So the goal of the project, our ultimate goal would be that the protection then would last for 2 weeks instead of a few hours. So what are the bases for that? We know already from the literature and work from my colleague Niels Hoos that people will smell different from each other. And here you see a sample size of 48 individuals that attraction to Anopheles was tested. And what these graphs basically show is that there are people that are very attractive to mosquitoes and others that are not, poorly attractive. Not attractive is almost impossible, but they're more to mosquitoes. And you all know that. You all know someone who is very attractive to mosquitoes and others who are less attractive to mosquitoes. This has been also correlated with their skin microbiome. So we know that people that have a higher diversity of bacteria on their skin, they tend to be less attractive to mosquitoes relatively to someone who has lower diversity of mosquitoes. And also some bacterial species have been correlated with being attractive or not. So a Staphylococcus is a species of skin bacteria that are correlated with someone who is very attractive to mosquitoes, while Pseudomonas species are correlated with people that are less attractive to mosquitoes. So the ideas we publish in this opinion paper in Trends, we think there are many ways to go if you think of using skin bacteria to protect us against mosquito bites. So the first route on the top of the slide, we think that, for example, we could try, for example, to increase the relative concentration of a bacterial species that is characteristic of a poorly attractive individual, and therefore making that individual less attractive to mosquitoes. On the second route, what I talked before about increasing biodiversity. So there are also indications that people that live in rural area relative to people that live in cities also have a difference in biodiversity of skin microbiome, maybe related to how much cosmetics they use and so forth. So increasing biodiversity on your skin could also be a way to go. And the third route, see that maybe not so clear from the drawing, if I don't add the words to that, we want to also try to bioengineer. So bioengineer, so modify this bacteria to, for example, start producing more of a repellent, and that could be even inspired on the plant-based repellents that we use today, but with the advantage that they could be produced continuously by the bacteria. So my project is also very much focused on this bioengineer, and we have a company that actually is a partner, what helps to eventually not have the findings only in the literature, but actually getting them into the market. So we have Ginkgo Bioworks working with you, and they are responsible for the bioengineered of the bacteria, and then we test them for mosquito attraction, and we also collect the odors to do the characterization of the odors to see if they really producing what they bioengineered to produce. And we also have a collaboration with AZITRA, and AZITRA is also a very important piece of the project in trying to eventually try to put this in the market, because AZITRA can also do the clinical trials on animals and in humans. And AZITRA has already the know-how and this whole idea of the probiotic or the microbes that for topical application is already being used for other skin diseases, for example, for psoriasis and eczema. So they have the know-how, and it's good to have them for the eventually doing the trials when we succeed with this. So here again, the three routes that I told to you, and we are focusing on routes B and C. So the bioengineered part, but also the increasing biodiversity, and I'm going to show you some results of that. So we are working actually not only on Anopheles, so not only on malaria, but the goal is to find something to produce a probiotic that would repel also other mosquito species that are responsible for transmission of dengue, malaria, and chikungunya, for instance. We have like 120 individuals for which we test attraction of human odors to the different mosquito species, and we also collect odors of 120 individuals in our study. And the goal is to correlate mosquito attraction with the or human odor signatures, so we know which odors we should target that are produced by the bacteria, and make them produce more or less of these odors. Of course, this is a challenging project because these odor blends, they are very complex, and we have it's not for sure that we are going to find odors that are that could be used as a repellent, or that we could be used to reduce attraction to all the 4 different mosquito species we are working on. But if we look at the literature, for example, and this is not based on our study, but this is basically on literature review, every data that some of you have collected already and that people have collected, there are compounds that in common that are, in this case, I'm showing only attractants, but there are compounds in common that are attractants to mosquitoes, and also to the 3, in here I show you 3 mosquito species. So Aedes aegypti, Aedes albopictus, and Anopheles gambus, and these are the 13 compounds, for example, that are commonly used by mosquitoes to locate us. So skin bacterial volatiles. So we have designed some in vitro models to study these volatiles that are produced by skin bacteria. And we started small with increasing complexity. So we start with a single bacteria species that is very abundant on the human skin. And then we have models with 2, 3, and 4 bacterial species. We also have models, in vitro models that resemble the composition of skin composition of a poorly attracted individual, of a highly attractive individual, and we studied that to understand what kind of odors they produce and how is the attraction to the different mosquito species. See, I'm sharing one slide on the results of this. So here, these data clearly show you that in the first block, for example, that if you increase complexity, so increase the number of species of bacteria, you decrease the relative attraction. And in this case that I'm showing is Aedes aegypti, the response. We also confirm that in the models we have for a highly attracted individual compared to the model of a poorly attracted individual. We also confirm that indeed, an individual that is highly attracted to Anopheles gambus, that's what we based this composition on, is also highly attracted to Aedes aegypti. So that gives us hope that we will find compounds that can be useful for more than one mosquito species. So yeah, to summarize, we are working on this, especially on route B and C, so especially increasing biodiversity and on the bioengineering path, but we are also working on the animal side also to protect animals from biting insects in general, no mosquitoes, but biting midges. And then we will also work on increasing the relatively concentration of a bacteria that makes make an animal less attractive to mosquitoes. Engineered part, we basically focus on one bacteria, and then we like to think about this concept of masking repel. So we want to reduce compounds that are attractive and increase compounds that can repel mosquitoes at the same time. So we like to think it this way in terms of mask and repel at the same time. And we use one main bacteria species that is very abundant on the human skin in general. So what I would like you to remember from this talk, that our skin microbiota play a key role in mosquito attraction. Manipulation of the skin microbiome is already a possibility and has been used already to treat skin diseases such as eczema and psoriasis. And the bioengineering part is just one of the ways to go, but there are different paths that we can use to use microbiota as a topical probiotic. With that, I would like to thank you very much, and also thank you many people that work with me in this project, but especially the people that help to get the work done, like Carly and Julian, and also my colleague Niels Verhoost. And once more, thank you so much for the attention.
So, Danny, a fascinating presentation, and thank you for that. I think all of us found it really absorbing background, and clearly this is a very productive and appropriate direction of research. We wish you well. I get the impression it's still fairly early days. You're in the early research stage. You're onto some very, very good ideas, and hopefully it will lead to something that alleviates the frustration many of us have currently with topical products that work very well for 1 or 2 or 3 or maybe even 4 hours. But even the gold standard DEET, really struggles to maintain its full effect after 8 hours. So it's that frustration of people having to pay for a product, they apply it early evening, but then it loses its efficacy, towards the middle or early hours of the morning, and then you get a biting peak again early morning, and you're no longer protected. So we wish you well in your research. There are some questions that are likely to be posed to you, and thank you for your time. So I'm going to move on now and introduce our next speaker, who will be Dr. Chris Dayun Kim. Chris is a postdoctoral research fellow in the Department of Entomology at Kasetsart University, Bangkok, Thailand, in the department that is headed by the well-known and respected Professor Tirapap Charionviriapap. Chris majored in chemical engineering for his first degree, and then moved to the Urban Entomology Laboratory at the University of Science in Penang, Malaysia. He did his PhD at Kasetsart University under the supervision of Prof. Tirapap, specifically to evaluate cost-effective spatial repellent tools to address outdoor transmission of mosquito-borne disease. And Chris is going to talk on novel chemical lures, attractants, for Aedes, Culex, and Anopheles. So Please take note, this talk is not on repellents, but I think you will agree that the subject of attractants is equally important, as they are the basis for many of our vector surveillance methodologies, but potentially also as part of mass trapping to reduce mosquito populations, and also as lures to things like toxic baits. So we thank you for making yourself available, Chris, and we look forward to your presentation. Thank you, Chris. Over to you. Please activate your video and also turn on your audio, please, Chris.
Thank you very much for your kind introduction, Dr. Ryo. This is Chris Dae-yoon Kim from South Korea, who is a current postdoc researcher at the Kasetsat University. I'm working on the mosquito spatial repellent. And today I will give a short talk about my PhD research topic, developing the chemical lure product for the mosquito surveillance tools. So it is sounds like opposite for the repellent and attractant, but actually it is linked. We have to get the knowledge, understand of the mosquitoes' behavior and responses, and also the chemical sciences. Just now, Dr. Danny gave us very interesting story about developing their odors from the human body and engineering the microbiome. And today I will show you how we test that kind of the cooked chemical in the laboratory and also semi-field. Let me start. There is a tool to testing the pesticide and repellent to evaluate the efficacy of the toxicity or contact irritancy, and also spatial repellency, so-called high-throughput screening system, which is developed by Dr. Glico almost 20 years ago. And later on, the WHO guidelines recommended this device to testing the efficacy of the spatial repellents, along with the wide tube or olfactometers. Recently, 2018, WHO guideline published for attractant evaluation for the mosquito surveillance traps. However, in this guideline, they recommended only for the wide tube olfactometers, but they didn't mention about the HSA, but they referred to the previous guidelines, which is for the spatial repellent. So my supervisor and my co-advisor asked me to make one more function of the HSA, which is for the evaluating function for the attractant. So this is how my PhD research has started, to add this device one more function, which is attractant. As you can see on the screen, this device is very simple. It consisted with the same size 3 chambers. And the screen showing that the red means treated, the green means untreated with the chemical, pesticide, or attractant. In this case, attractant. So we baited in the red color and we unbaited in the green color. Let me talk about this device. There is a small entrance between chambers, so-called the butterfly doors. So basically, we put the close position at the first stage, and then we introduce the 20 female mosquitoes, which has not blood fed, but 3 to 5 days ages, into the middle chamber. After that, as we introduce the mosquitoes, we have to give them 30 seconds to be calm down. After 30 seconds, we open up the butterfly doors for 10 minutes for the mosquitoes choice study, so that the mosquitoes can freely fly inside their chambers. So if the attracted baited showing attraction, then the mosquitoes will move to the lead chamber. But opposite side is untreated one. So if the attractant candidates repel the mosquitoes, then they will move to the unbaited chamber. After 10 minutes, the door again and counted the number of mosquitoes in each chambers to calculate the percent attraction showing using the formula on the screen. So we published 2 papers from this study to add the attractant evaluation function. So the first one, we optimized the busy lure, chemical lure using the heat assay, and then we screened the newly developed Kasetsat University lure using the heat assay. For more details, please type the diffusion assay for the mosquitoes in the Google. However, as Dr. Danny already explained us about the difficulty of the cooking the attractant or repellent from the different blend or chemical mixtures within various of the proportions and ratio, even though the candidates showing very, very good performance in the laboratory, it is only the left side study. So we have to repeat it in the semi-field screenhouse assay in the larger scaled field study. So objective of this study is the semi-field evaluation of the novel chemical lures. As you can see in the picture, we have the semi-field facility in the forest. And this facility covered with the fine measured metal screen so that the mosquitoes can go out or cannot go in, cannot escape from the inside. As late Dr. Michael Banks mentioned, the beauty of the semi-field screenhouse assay is we can test the actual mosquitoes' behavior in the natural condition under the controlled and known mosquitoes biological conditions. So we can test the repellent or attractant in the semi-field before we move on to the real outdoor field test. Actually, we established this facility 10 years ago, and one of my senior, Dr. Salazar from Philippines, he studied Aedes aegypti's density, mosquito density, and also the trap density in the semi-field screenhouse to see the optimal number of the traps and mosquitoes for his testing. So he tested BGS traps baited with the busy lure. However, he didn't compare with and without the lure baited busy traps, but I could get some idea for my study regarding the density of the mosquitoes and also traps. And last year, there was another study from one of my senior, Dr. Chu Tifeng from Kasetsat University. He tested spatial repellent, which is the transfluorine treated plastic sheet using the human volunteers. So as you can see on the screen, right-hand side, the control collector didn't apply the chemical, non-treated, but the other side, the collector having the treated plastic sheet so that the Aedes minimus lilies in the middle can make a choice whether they repel or they just ignore the chemicals. So basically this semi-field study can demonstrate the natural movement in the screen enclosures. So for my testing idea, I just follow this method, but change their volunteers to the BGS traps and also black hole UV light traps. So this is the recently published paper about the semi-field evaluation of the novel chemical lures for diurnal mosquitoes, Aedes aegypti, and 2 nocturnal species of mosquitoes, Culex quinquefasciatus, and also Anopheles minimus. However, as the novel chemical lure was haven't tested in the semi-field, we had to standardize the semi-field assay using the lure unbaited BGS traps as a preliminary study. After that, based on that information, we could conduct the optimizing and also screening assay in the semi-field using the lure-baited BGS traps, which is embedded BG lure or carbon dioxide or also Kasesa University lures candidates. And finally, we tested the enhanced performance of the black hole UV light trap by adding our selected KU lure candidate. This is the traps I used, BGS traps for the experiment number 1 and black hole UV light traps for the experiment number 2. For these experiments, I used the Kasesa University lure candidate, which was cooked by one of my co-advisor, Dr. Tirachat Limparsat from Department of Chemistry from Kasesa University. Let me introduce the semi-field study settings briefly. As you can see on the screen, I put the 2 BGS traps at the end of each side and released 50 female mosquitoes, which has not blood fed. And I baited one BGS trap and unbaited the other one. This is the actual picture in the semi-field. After the testing, I counted the number of mosquitoes from each traps to calculate the percent attraction using this formula. And following this protocol, I tested one pack of BG lure, which is positive control, and another positive control, I added carbon dioxide. And of course, we screened and also optimized the amount for the semi-field assay using the 6 different KU lure candidates. For this study, I tested Aedes aegypti during daytime and also tested Culex quinquefasciatus during nighttime. Let me introduce one by one. This is the result from the Aedes aegypti during daytime from all the lure candidates. It's quite busy table, but please look at this part, the number of mosquitoes attracted by the unbaited BGS trap and also baited BGS traps. And I compared between 2 to see the significance. As you can see in the red box, 0.1 gram and 0.5 gram of the KU lure number 1 showing the significantly higher number of mosquitoes in the KU lure number 1 baited BGS trap. Using these numbers, I could calculate the percent attraction and the percent attraction also showing the positive, which is attractant, whereas the other candidates showing negative, which is repelled. For the positive control carbon dioxide, it also showing significantly higher number of mosquitoes baited into the baited BGS trap, so that the percent attraction also higher, but it was not significantly different with Kasesa University lure number 1 for the Aedes aegypti. And I conducted the same protocol during nighttime for the Culex quinquefasciatus, and this table having the same format. As you can see in the red box, KU lure number 6 baited BGS trap during nighttime attracted significantly more number of Culex quinquefasciatus, so that we got the highest percent attraction score from the KU lure number 6. For the positive control, carbon dioxide showing significantly more numbers as well in the baited carbon dioxide baited BGS traps. However, the percent attraction was significantly higher compared to the KU lure number 6, which was a bit different with the diurnal Aedes aegypti, so that we can see some kind of the species-specific different responses. Actually, this kind of phenomenon attracted by carbon dioxide in the outdoor for the nocturnal mosquitoes has confirmed by one of the researcher in Thailand, Dr. Pachara from Mahidol University. They published one of the paper testing CDC light traps baited and unbaited dry ice in 2015, and she concluded that by adding the carbon dioxide, it significantly increased the capture rate of the Anopheles species. However, she couldn't see the significance for the Culex species because this study conducted in the rural area near the Myanmar and Thai border, so-called Dak province. So she added in discussion that the previous studies also actually demonstrated the power of the carbon dioxide for the CDC light trap, which attracted the most for the Culex quinquefasciatus. Now, I also wanted to see whether this kind of selected lures can enhance the trap efficacy, so that we tested the selected KU lure number 6 for the diurnal nocturnal mosquitoes, which is Culex quinquefasciatus and Anopheles minimus, using the black hole UV light traps. And the protocol was the same with the BGS traps. So this is the result for the nocturnal species. As you can see here, the Culex quinquefasciatus attracted by the KU lure number 6 baited BG black hole UV light traps, so that the percent attraction was also increased significantly compared to unbaited black hole UV light traps. However, we could not see the significant from the Anopheles minimus, even the number has increased. So we can conclude that for the nocturnal species, Culex quinquefasciatus, whether the trap has light source or not, for the both BGS and black hole UV light trap, by adding the Kasetsat University lure number 6, we could see the significantly enhanced trapping efficacy from the semi-period study. Conclusion. This is the figure from the previous study from the Hitch assay conducted in the laboratory. And you can see in the blue box, the KU lure number 1 was selected as the best lure candidate for the Aedes aegypti. However, for the nocturnal species, KU lure number 6 increased the most highest percent attraction. So actually this result was repeated in the semi-period. So this study in the semi-period, we demonstrated the accuracy of the Hitch assay from the laboratory. However, as I show you the result, I could not see the significant enhanced trapping efficacy from the Anopheles minimus in the semi-period screenhouse assay. So we need to do the further trials in the open field to see the efficacy of the KU lures. And this is the last slide for the methodology for the developmental process of the chemical lure. As you can see, we have to repeat it from the laboratory study to the semi-period and also open field. And all the feedback from open field should go back to the laboratory to enhance the performance of the potential lure candidates. So this is what I prepared for today. I would like to thank you for my funding sources, Thailand Research Fund and Graduate School of Kasetsat University, and also the KU Research and Development Institute, KODI. And especially thanks to my co-supervisor, late Dr. Michael Bang, for his contribution on this study. So hopefully this talk could be one part of his memorial talk. And also thanks to my supervisor, Professor Tirapap Charyonbiyapap.
I take it you've finished your presentation, Chris, which was fascinating. Clearly, you and your colleagues at Kasetsat University, you're very well rigged up and set up to do some very practical and useful experimentation on repellents and attractants under insectary, laboratory, and semi-field conditions. So we wish you well. Very interesting results that you are coming up with there, and very interesting presentation. Personally, I'd like to go back to your presentation and more slowly go through it and look at those results. My brain just works a bit slower, so I need to spend a bit more time, but very interesting. Thank you for that, Chris. There are a number of questions already coming in, both for you and for Danny. We'll get to that in a few minutes. I'd like to encourage our audience members, if you have questions, pop them into the Q&A box, and hopefully we'll get to them. We have limited time, but we're going to try and at least pose some questions to our panelists. Our final speaker will be Thomas Putzer, who is a senior group leader in the office of the chairman and CEO of the company SC Johnson & Son. Tom holds a Bachelor of Science degree in limnology and an MBA from the University of Wisconsin. He first worked at Kimberly-Clark, leading product research and launching in more than 30 developing nations, and then joined SC Johnson in 2008. Tom works with a very talented group of people in the global public health space to create social impact for the base of pyramid. And the base of pyramid is basically those 4 billion plus people globally that earn less than a few US dollars per day. So Tom is going to tell us about his work with communities in sub-Saharan Africa and also Southeast Asia. Thank you for joining us, Tom. We appreciate it, and we look forward to your presentation. Please go right ahead, Tom. We look forward to your information. Thanks.
Sounds good. Thank you, Dr. Leo. I appreciate the kind introduction there. And thank you also to Dr. Danny and Dr. Chris. I think it's the work that you're leading, the type of work that you're leading, that really serves as inspiration for us at companies like SC Johnson that try to develop new approaches and new innovations. So I'm learning from you as I listen here too. And Dr. Chris, it was also nice to see Dr. Bangs referenced a few times. I think his spirit lives on for sure. So I'll speak a little bit about what we're up to from an industry perspective. I will try and share what we're doing in this space and what we're trying to do to advance a few new ideas and maybe a different approach from a slightly less technical perspective. So a few key points that I'll focus on: who we are, what we're up to, and why SC Johnson is in this space. How we're guided by the people we're trying to serve, and some tools that we put out there to help prevent mosquito-borne disease, including repellents. So I imagine some of you may be familiar with SC Johnson. We've been around for a while, about 130 or so years. We've got sales in just about every country and operations in about 70. We're the leading manufacturer of household pest control products, and we operate one of the world's largest private urban entomology research centers. And I'm guessing some of you on the webinar here may have worked with some of our entomologists there, like Dr. Maud Meyer or Dr. Tom Mascari too. And they've played a strong role in some of the work we'll talk through. Within SC Johnson, we've got a group called our Base of the Pyramid group, and we're focused solely on disease prevention, primarily in the mosquito-borne disease space. The world's poorest 4 billion people, the base of the pyramid is a pretty big niche market. We realize that, but we're focused on trying to help those that might be the most vulnerable or the most at risk. There's a few key tenets of this group that we stick to, but one of them is this group, our Base of the Pyramid group, is not focused on making a profit, or is not focused on sales. It's focused on impact in the sense of people reached or disease prevented. We realize too that it cannot be sustainable if it relied solely on philanthropy. So we try and approach everything to create economically sustainable solutions, which is probably just some big words to say that we're trying to make sure that others are able to profit along the way, whether it's distributors, whether it's community health workers, whoever may be involved, so that it's sustainable beyond just SC Johnson. Like any good group, anyone that hopes to have meaningful impact, we've got a lot of strong partners behind us and with us. We know what we're good at and we know what we're not good at. And I imagine some of you on this call too are also represented here, but we learn a lot from working with all of our partners. And obviously something like malaria or like outdoor biting is big enough that it takes more than just one company to address it. It takes all of us. And I think at the core of what we do, we're really focused on human-centered design. And it's been eye-opening for us, I'd say, as a company, especially a company that prides itself on its technology and prides itself on innovations and coming up with wonderful solutions to prevent disease. It's been a little humbling for us to go through this process over the past, I'll say 10 or so years, but we really try to immerse ourselves in the lives of the people we're trying to serve so that we can develop solutions that fit within their lives. And this is just an example of one of the many projects we've worked on, but spending some time in Myanmar and Thailand to try and address outdoor protection in the greater Mekong subregion. And as part of that, we spent time, tapping rubber trees with migrant workers, going community to community, midwives, spending time with health officials or village leaders, or sleeping on the floors of homes to really try and understand what the people were trying to reach, how they live their lives and what's important to them. And ultimately the goal is to try and lead us to something that results in ease of transport, of course, something that's easy to get around, especially to the last mile, ease of implementation for either putting into place in a home or for a health professional to try and educate end users or people on this, and then ease of use, something that you can just maybe set it and forget it, or is just so easy to use you don't even need instructions. So this is kind of what we try to shoot for. I think along the way too, some interesting learnings, when we take a disease like malaria, it's been very, again, humbling for us. We've had people tell us, community members tell us, you get malaria from eating certain foods, or you get it from walking too much in the sun, or we're aware of malaria, but it's just the way of life. It's just something we deal with. And this has really forced us as a company to guard against our hubris a bit. Yes, we're trying to do something meaningful, and yes, we're trying to prevent disease, but it's perspectives like these that really kind of ground us and help shape the solutions we come up with, in that malaria may not be the focus for the people we're trying to serve. And so how do we develop something that fits more seamlessly into their lives? To go a little deeper there and take someone like Kotin here, this is a rubber tapper in Myanmar that we spent some time with, and he had mentioned too, I don't bring any mosquito repellent with me because it's too expensive and bothersome to use all day. But you know what? I blow some smoke onto my body to repel mosquitoes. It works for 5 minutes, but it's not that effective. But since I smoke anyways, it's easy to do. And there's a lot of this simple quote, the realization that, I don't think this blowing smoke on myself works, but I'm doing it anyway. So I'm smoking anyway. So I'm just going to do it. It fits into my life. Or the fact that he said, I don't use any repellent because it's too expensive and bothersome to use all day long. I'm sure many of us have heard that too. And let's just focus on that point a little bit, the all day long part of it. one of the projects we worked on in partnership with WWF in Cambodia looked at people like Coteen, like migrant workers, and in this case looked at rangers and their journey. What is a day like for them? What is a 24-hour period like for them if they are concerned about having to use something all day long? And it was interesting, and again, this may be a pretty salient point or a simple point, but just looking at the periods of high risk mapped out against their activities hour to hour really helped convey to us that there's times when they're at high risk where they're protected, where they're sleeping in the forest under their hammock nets, for example. But there's times when there's high risk, like when they're cooking or eating or when they're walking into the forest, that they really need to be protected. But I think a key learning here is that we don't necessarily, in this case, need to focus on protecting the entire day or the entire 24-hour period for mobile workers. We just need to focus on protecting them during the time that they're at high risk and not protected by some of the core interventions like mosquito nets. This is just one perspective, but I think this is, the human-centered design approach has led us to learnings like these that guide not just the technology development, but more so the communication around the interventions we're putting out there. And I think this is a little summary of this, personal repellents as they exist right now, not just the attributes or the usage, but really the education around them can be tailored to situations where residual transmission exists and existing interventions may be constrained. So although again, we're a very technology driven company, and this is an APMEN Tech Talk, one of the things we really focus on is trying to understand the people we're trying to serve and tailoring the communication and the education around the possible interventions accordingly too. All that said, we're definitely interested in the work that Dr. Danny and Dr. Chris are driving as well, because those are some of the new technologies and approaches on the horizon for us too. Just really quickly too, I'll speak to the spatial repellents. Dr. Chris, you mentioned Dr. Greco too, and we're working with him and Dr. Nicole as principal investigators along a path here to try and garner policy recommendation for spatial repellents. We're also in partnership with the Gates Foundation and Unitate, and I think many of us know how this goes. It's a very long, very expensive journey, and we're a ways through the process here. Again, Dr. Maud Meyer has driven a lot of this too, but we're in the part of the process here where we've got trials that just kicked off in Kenya and Mali with this intervention, which I think may even be very similar to what you used, Dr. Chris, for your the vest with the plastic sheets. This is something that lasts up to 30 days, and we're already working on the next iteration of that, which should be available about the same time too. But we've got products that are lasting up to 6 months and a little bit longer here too. But we're on this journey here to try and garner recommendation for these interventions. And although they're not targeted solely for outdoor protection, it is something that people can use in a kind of a mixed indoor outdoor setting, as long as they're not moving too fast. Just a quick summary slide of some of the different repellent type products that are out there. And if I'm really honest, I hesitate to show this only because I don't want it to come across like I'm trying to sell product, and that's definitely not the case. Again, we donate a lot of this and don't try to profit off of it. But I've just listed some different comparisons here for price comparisons for each of these interventions and some of the different benefits and duration. And I saw one of the notes in the chat too, in the Q&A. We can definitely make this available along with some other resources that provide more detail around the different types of repellents that are out there, or at least that we provide out there. there's a number of different repellents, and many of them, at least on the personal repellent side of things, are leveraging DEET or picaridin as the active ingredients. But again, we're happy to provide more information in the chat or following this webinar. Just a quick summary slide too of some of our highlights over the past year or so, and we've done a lot of work and reached a fair amount of people just in the past year or so, reached a good 20 million or so people. And I mentioned we're focusing on education is a big part of what we do, not just the technology, but the education. And that's probably going to be a good focus of what we do going forward too. So I'll keep it short and just stop there and say thank you. I'm happy to answer any questions.
Okay, Thomas, thank you for that overview. It's very different from the kinds of presentations that both Danny and Chris presented. Here we were starting to delve into the practical aspects amongst user groups, primarily those in rural areas where malaria is sustained and where poverty-related issues drive them into activities and livelihoods and living conditions where they're at higher risk of exposure to vector mosquitoes. And you refer to some of those issues, which is, it's completing the picture from research through to the more practical field aspects. Thank you for those perspectives. So we've got 200, at one point it was around 220 participants and 18 questions as far as I can see. So not all that many questions. A good number of comments in the chat boxes. I've not been following the chat box all that well, but the few I've seen, very great, useful comments to read through. So just going to the Q&A box, again, thank you so much to our three panelists. Genuinely appreciate the time you spent putting together your presentations, and also to our audience members for setting aside time to join. So here's a question. It's not really I'm not sure if it's a question, Michael McDonald, or whether it's an observation that we need to remember there's a difference between a true repellent substance as opposed to a product that merely masks, the attractant chemical attributes that lure mosquitoes to our bodies. So there's that distinction that needs to be made between repellents and masking products. I'm going to ask Danny if you have any comment on that aspect, but there's another question here that relates to human blood groups and the possibility that people in different blood groups may be more or less attractive to mosquitoes. Danny, do you have any comments on that?
Yeah, sure. Yeah, well, with respect to attractants versus repellents, yeah, sure, these are different things. And in our project, we think it's important to think of both together. So we want both to reduce attraction, so mask and repel at the same time. I think also because, yeah, I think resistance over time. So if you have more targets at the same time, you can think of imagine a project that's more durable in time. And with respect to blood type, as far as I know, there are no evidence that is correlated with blood type, that attraction to mosquitoes is correlated with blood type. I would argue more, again, that is more about the skin microbiota. And again, there is no correlation between skin microbiota and blood type as far as we know.
Okay, don't go away yet, Danny. There are a number of questions that are directed at you. So here's one here, right up near the top of the list, one of the early respondents. So the question is how the research you're doing on the skin microbiota, how does it stack up against products like DEET? And you made the very, very important valid point that DEET and the other topical repellents, the synthetic chemicals that we're using have a limited duration because they evaporate, and that's the basis of how they repel mosquitoes, is they need to be odorized and reach the mosquitoes before they land on us. So, but in terms of efficacy in strength of repellent qualities, do you have at least any initial indication of the quality, the potency, if I can refer to it as such, in terms of the repellent effect between the skin microbiota and products like DEET? Any comment?
Well, I think the most honest comment is that we are not that far yet to compare relatively efficacy of the skin microbiota projects that we aim at developing. But some of the compounds that we see that are potential repellents and or that could be used as mask, they are known, but what we think is that they know already from different researchers, but what we think is that the powerful thing is to combine more than one and in one other blend, and this has been also shown before related to that, and that I think it makes it more efficient than to the compounds we have today that are mostly a combination of a couple of compounds only. Again, that they are going to be produced continuously, so that that's the powerful thing of using the microbes in that sense. Does this answer your question,
Danny? Yeah, no, that's great. Thanks, Danny. So here's another question here from Aulia. We're now moving to Chris. Moving to you, Chris, to Aulia Rahman, who raises this issue of carbon dioxide as an attractant and its relative efficiency in attracting vector mosquitoes to a trap. So, you know, comparison of UV light by itself and then carbon dioxide. So I think you've already answered the question in your reference to that particular publication, which showed the high levels of efficacy of carbon dioxide. And that, you know, for what it's worth, I can just say I've done a lot of light trapping in Africa, and routinely we found that the combination of UV light with CO2 in the form of dry ice or so on is just a very powerful way of drawing vector mosquitoes to your trap, as opposed to pure UV light by itself, where you get overwhelmed with other insect populations, but not all that many mosquitoes. But Jacques, don't go away, Chris. Jacques Charlwood made the comment here that bacteria, these are his words, bacteria around the feet are especially attractive. Why not test mosquitoes instead of hands in your tests? And that's, you know, there are published results on this that show that ankles and feet are you know, far more attractive to malaria vectors, both in Africa and in Asia Pacific, than other parts of the body. And in fact, if you take a plastic bag and you cover your ankles and feet, most of the, I say most, it's a deliberate choice of words, most of your malaria vector mosquitoes that would have come to bite you if you were sitting or standing outdoors will move to another person rather than find a spot higher up your body to go and bite. So there's a very strong preference for ankles and feet. And yet, you know, in our insectary and laboratory trials, we keep on sticking our hands and arms in these arm in cage trials. And I suspect it's more for practical reasons, although we know that ankles and feet are actually more effective. Do you have any comment on that, Chris? Why would you prefer using hands in your trials than feet? Any comment?
Thank you very much for your explanation about the questions. Actually, for my testing assay, we do not use the human itself. We just use the assays, which has not human involved. But for your question, why the mosquitoes, even though they prepare much more to feet, but we are testing hands. But as far as I know, there is another testing assay using the pit. So I think, well, it is my opinion, but the assays could be chosen depends on the purpose and also depends on the mosquito species, because each species has their own species specific preference. For example, for the Dr. Danny already explained to us, they attracted by different formulation, different number of mixtures of the same for my study. When I test the KU lure blend, which is mixed with different kind of chemicals and also different concentration, it obviously significantly showing different percent attraction. For example, octanol, which is very commonly used and selling in the market with the cheaper priced, the UV light trap, their companies are selling together with the octanol attractant. However, for the octanol actually came from the human sweat odor or body odor, and it most the Aedes aegypti compared to the Culex quinquefasciatus, because this Culex quinquefasciatus prepare the other host, which is bird. They do not release the kind of chemical octanol. So when I test the different proportions of KU blends with the lower concentration of octanol, it was much better to attract the Culex quinquefasciatus. So again, we have to consider what is the purpose of the testing and what is the target mosquito species. So for your question, Dr. Leo, why in hand rather than using the pit? They're also using the pit, and there is the assay protocol for using the pit as well.
Okay, thank you, Chris. Sorry, while we have you, there's another question here from Jacques Charlwood. Just interested, you don't have to go into detail, just an off the cuff, yes or no. Have you picked up any differences in colonized insectary mosquitoes in terms of their responses to attractants or repellents and so on? Because, you know, for very obvious practical reasons, we tend to go to insectary colonies to do our trials on specimens of known age and known confirmed species, etc. But have you picked up any differences between colonized specimens and wild specimens? And is there a need to be careful about these differences?
Thank you for your following question, Dr. Leo. Actually, in my first publication using the HITCH assay in the laboratory, I compared the susceptible strain and also the pyrethroid resistance population from the field site to see any differences regarding in the host-seeking preference or behavior. But I could not see the significant differences between two strains and population. However, even though we couldn't see the differences, we need to conduct the semi-field study using the field population, because the different geological geobiological differences should showing something different. But from my test using the lure, I could not see any differences for these two laboratory and field population.
Chris, thank you for that. So here's sorry, I've just been skimming through the questions that have been posed here, and I'm very conscious that we haven't come to you yet, Tom. So I try to find a question that we can pin you a bit, pin you down. So here's an interesting one.
No worries, no worries.
The question is from Lech Ignatowicz. How do you deal with a situation when only part of the community can afford repellents and mosquitoes? And so these mosquitoes are pushed to the other non-protect. I mean, this is an issue. This opens a whole can of worms that, at least my impression is, WHO is very lukewarm in its in considering repellent products, because it doesn't really deal with, you know, overall reduction in malaria case numbers. You prevent the mosquito biting one individual, but you're simply causing that infected mosquito then to go and bite another individual. So repellents make a difference at individual level, but not at a population level. Your transmission still occurs and your overall malaria numbers may remain the same. You're just shifting the transmission to different individuals. So do you have any comments about this issue of repellents cost? There's no one out there willing to cover the costs of free handouts of repellents to community members, as far as I know. So communities have to pay for repellents using what little money they do have for, you know, casual expenses, well, for voluntary expenses. How does one deal with that aspect, that it's one of the few things that we can really apply for reduction in outdoor transmission, but it's a problem for communities? Any comments on that, Tom?
We could do a whole nother webinar on that. That's a great question. There's so much wrapped up into that, and I love it. I think, so I'll answer it a few different ways, but the first to say, yeah, it's a real issue, right? And it's part of why WHO recommends personal repellents for personal protection as opposed to community protection, right? I think there's a few ways we're trying to address that, or a few ways we look at that. Like, I mean, personal repellents, skin applied repellents, they shouldn't replace something like mosquito nets, and they probably won't ever replace it, right? You know, like, there's core interventions out there that work. I think something like a skin applied repellent is appropriate for those residual transmission times, or, you know, the times where people are not under their nets, you know, the areas where they're still at risk. So it's not like they need to be protected all day by them. I think as far as what we're doing, and yes, there's expense by it and behind this, and it's asking a lot of people to pay for these things. While at the same time, can we rely on straight up philanthropy? We're going through the process of getting repellents PQ listed, pre-qualification listed through WHO, which in turn, in theory, would open up funding through global public health channels. That's a big if, but we're going down that road in terms of a funding stream, and then also in terms of making them available. I mentioned actually right before this call, we're sending some to CNM in Cambodia and the ministry there to try and make the repellents a little more available. We realize that philanthropy will help a little bit, but is it something that's economically sustainable? We'll need a few different approaches. So that's a little bit of rambling. There's a few different areas, a few different ways we're looking at that. But yeah, happy to tackle that in the next webinar too, if you want.
Yeah, it sounds like we'll need another webinar. Lots of questions still to be asked. So here's a quick one for our chemists, Danny and Chris. I'm going to leave it up to either of you to respond to it. The question is, it relates to, I mean, we know about development of resistance within mosquito species to insecticide-treated bed nets and so on. How about development of resistance to repellents? Are there any examples that you can quickly rattle off where mosquitoes have been shown to become resistant to repellent products? Chris, you were nodding your head. Do you have any knowledge of that?
I heard that about the question why did not.
Chris, you've frozen.
Function or mechanism.
Carry on. Carry on.
Yeah, so it is because the different mode of action or different physical mechanisms between side and DIT. So I think that is why the DIT resistance for the mosquitoes.
Yeah, thank you, Chris. Danny, do you have anything else you'd like to add to that?
Well, I think resistance is a possibility always if you target one single compound. So the way to go is to have a more complex blends to avoid that along the way. That would be my thoughts.
Sure. Yeah, so I mean, this whole issue of outdoor biting and how to address outdoor biting is a highly frustrating. You know, we've had so many wonderful innovations over the previous century, and we just seem to be hitting a brick wall and bumping our heads on this issue of reduction in outdoor transmission. And, you know, people are saying, okay, there's attractive targeted sugar baits and so on. And there's insecticide treated, ivermectin treated cattle, and, you know, insecticide treated sheets that you hang outdoors, various ways. There's also larval source management. You know, those are targeting reduction in available population, mosquito vector populations outdoors. But there have also been wonderful trials conducted in Africa at Ifakara and elsewhere on transfluthrin impregnated materials, bands or strips that you can wrap around a particular space. It acts like as a spatial repellent to protect people within the area, the sphere of influence of those spatial repellents, and attaching a piece underneath a chair or taking an transfluthrin impregnated strip and sticking it onto a sandal or something like that. So, you know, people are trying out different things. There's another one I'd like to mention, and I think Frank Langeveen, you're actually in the audience, or you were earlier on. Frank is trialing a product called Maya. It's a topical, it's a soap, basically, which incorporates DEET. And it's a very pleasant smelling soap. And it's particularly appealing in West Africa, where cultural practice is that women and children go and bathe, small children go and bathe late afternoon. And they use this wonderful soap as a cultural practice. And now they've just incorporated the DEET. And this seems to be helping reducing malaria transmission in under 5-year-old children. And of course, that's a particularly susceptible group, target group, that suffers very heavily from malaria mortality and morbidity. So, you know, we just have to keep on trying. I'm going to open it up a bit and ask, is there anybody in the audience? This is a big issue, people, this whole issue of outdoor transmission. And you know, we kind of, I get the impression we're floundering around. Is there anybody that wants to philosophize, speculate as to which direction we should be moving in? Where are the promising aspects or technologies that we should be focusing on? If any member of audience, if you would like to spend just a minute, raise your hand, and then I'll ask our technical experts to give you an opportunity to go live and give you a voice, as it were. So anybody with any opinion on what should we be doing about this outdoor biting aspect? Stick up your hand. Dr. Pon, is that possible?
Yes.
Okay. Anybody willing to stick up your hand, stick your neck out? In the meantime, there's a question here from Emmanuel Forlack. What is the difference between body odor and body flavor? So unless I'm missing something significant, body odor is a smell, body flavor is something, the mosquito has to bite you to get the taste. And that's very different. You know, the mosquito has to be already in contact with you and biting you, whereas the body odor emanates from you and reaches the mosquito some distance away from you. So very different modes of action. So have we got somebody?
Yeah, Dr. Leo, yes, he will talk about. So please.
Is it Lek? Okay. Lek, please give us your words of wisdom.
Welcome from Sweden, where we also have a huge mosquito problem. So I'm from Molecular Attraction. We work on the passive systems for passive traps, which are not using sugar. So just to how we see this is that it needs to be, of course, a collaborative effort. We would like to see that there's a very strong repellent system indoors. So the mosquitoes are actually pushed outside. And then our traps or any other passive systems can actually take them over outside the either huts or apartments. Frankly, humans are always going to be the best attractants for mosquitoes. So if we can just push them away a little bit from, let's say, the sleeping inhabitants of a hut, and the mosquitoes have nowhere to go but to the passive traps or passive systems. This is how we see. This is at least our approach, what we think could happen.
Yeah, Lek, I think, you know, there's a lot of truth in what you say, but I can't help but think, you know, it's not all or nothing. There's no single magic bullet. We're going to have to do this the long
oh God, no, that's just a piece of a puzzle.
Yeah, exactly. So, you know, a combination of methods. I like the idea, you know, for many of the vectors here in Asia, Pacific region, and at least for a couple in Africa, especially Anopheles arabiensis, that are strongly attracted to cattle. And in large areas of Asia, rural people keep cattle very close to the household. And if we can, you know, find some money to subsidize people to treat their cattle with ivermectin, you get much larger numbers of mosquitoes going to the cattle than to humans. And especially if the humans tend to be covered by bed nets for much of the night, you know, cattle are just sucking in large numbers of mosquitoes. And ivermectin treated cattle are very effective if treated, you know, at monthly or so intervals in killing large numbers of mosquitoes. But then, you know, you have to combine it, like you say, with different approaches as well. So I think we have time for one more question. Who would like to, Dr. Pui Pui, do you have a question you'd like to ask or an issue you'd like to raise? I see your hands up, Pui Pui.
No, Dr. Sharma.
Dr. Pon, are you saying we should give
Yes,
Dr. Rajendra Sharma a chance?
Yes,
Please come up, Dr. Sharma. Pleasure to have you on board. Please raise your question.
This is not a question, and what you said about this euprophylaxis or mixed sets, human dwelling as well as cattle set in a house, so that attract more mosquitoes, particularly in our country, India, Anopheles species. So that is already published so much work on these aspects. Number two, you are talking about extra domestic transmission, very difficult job, but as a personal protection method, surely we can interrupt the transmission in some areas which are highly endemic in Southeast Asia region. And one pheromone, that is a carryomones. So carryomones plays a very important role on human beings as what you are asking about this repellency or attractancy. So carryomones plays important role for different species in different endemic areas. So this is a chemical, that is a pheromone, carryomones, which plays important role.
Thank you, Dr. Sharma. Yeah, wonderful. So unfortunately, we've now reached our limit of 1.5 hours for this webinar, and I think I should bring it to a closure. It's been a particularly interesting topic, and I think the audience numbers reflect that. There was good interaction, good discussion, good comments on the chat, good questions. For all our panelists, for all our audience members, a very sincere thank you, Kopun Khap. And we'll be holding our next webinar in about 6 weeks' time. Feel free, if you have a particular topic that you'd like to suggest, please send it to one of us here at APMEN. In the meantime, there's a short poll. I'd be very grateful if you could take a minute or so just to answer the poll question there. But then thank you everyone very sincerely for your time. We appreciate it. Bye-bye now, everyone.
Thank you. Bye-bye.